So good morning students. Today in the last class we had discussed about the dihybrid cross. Okay. So that is very important. So once again I want to explain the dihybrid cross. Okay. So here you see dihybrid cross, which is also known as the inheritance of two genes. In which what will be the parents? The parents here we have taken two pair of contrasting character. What are the two pairs of contrasting character? Means that is round yellow crossed with wrinkled green. Okay. So here what are the alleles we have to take here? Means that is capital R, capital Y, capital Y. Capital R for round and capital Y for yellow. Crossed with the wrinkled green, that is small r, small y, small y. These are the dominant characters and that are the recessive characters. So during gamete formation, these genes which are present in the pairs, that is going to separate it. Here you see, these are the gametes which is going to be separated. Capital R, capital Y crossed with small r, small y. Then we have to cross this one to get the offspring of F1 generation. Okay, in the F1 generation, what we will get is that is capital R, capital Y, small y. So to this we can what we can call? We can call it as the round yellow because the small r and small y are the recessive characters. Okay, so here these are the whatever the offsprings which are produced, they are the round yellow types. There is no intermediate type of offsprings are produced, but they are similar to their parents. The next after this, after F1 generation, next step is the self cross. In the self cross, we have to cross this offspring with the same type of plant. Capital R, capital Y, cross with capital R, capital Y, small y. How we have to get the gametes over here? Means here you see capital R, cross with capital Y, small y. This capital R, capital Y, capital R, small r, capital Y, then small r, small y. This is about one pair. Here one more pair is there. We have to cross that also. Capital R, into capital Y, small y. Same thing you have to do here. You have to show this cross 2 times. Okay. Here there is no space there. You can show it beside that one. You can continue after this only. Okay, in the representation. This representation is very important. Okay. So here capital R, small r, capital Y, small r, small y. So these are the two parents to get the F2 generation. Now next we will see about the F2 generation. Why I am explaining twice means, why I am repeating this topic means because this is very important for 5 marks. If you represent this much, you will get the 5 marks. Now, lastly, during during F2 generation, the concept is very important. We have to put the chakra mode. Okay, for F2 generation. Okay, here now you see there are two parents. One side we have to take all these gametes. Okay, one side in a row we have to take all these gametes. In a one column we have to take all these gametes. See here, one is the male and one is the female. Male gamete is the pollen grain. In the pollen grain, it is all alleles will be present. Female gamete, that is the ovule, in which there is the presence of these all gametes. Capital R, capital Y. The next one, capital R, small y. Small r, capital Y, small r, small y. Then again here, capital R, capital Y, capital R, small y, small r, capital Y, then small r, small y. Now we have to cross here. See how means capital R into capital R, capital R, capital R, capital Y, capital Y. Okay. And next here to here. First term, listen here carefully. First term, first row, first column. Okay, this one we have to cross with this one first. Second thing, we have to cross this one with second row. Okay, capital R, capital Y, small y. Then with the third one, the same gene we have to cross with the third one. Okay, so here capital R, small r, capital Y, capital Y. Then with the fourth one, capital R, small r, capital Y, small y. In this sense, this completes about the first row and the first column. The next one, capital R, capital Y, small y. This one with this one, capital R, small y. Then this one with the third column, that is capital R, small r, capital Y, small y. This one with fourth one, that is capital R, small r, capital Y, small y, small y. Okay, then here capital R, small r, capital Y, capital Y. The next term, capital R, small r, capital Y, small y. Then small r, capital Y. Then small r, capital Y, small y. Then lastly, This one we have to cross with this one. Capital R, small r, capital Y, small y, and again this one with second one. Capital R, small y, small r. Then this one with third one, small r, capital Y, small r. This one with fourth one, small r, small y, small r. So this cross should be correctly and it should be mentioned clearly in the boxes. Okay, so you have to neatly draw this checkerboard, to neatly mention all the gametes. During the evaluation, they will see all these gametes. Okay, then next it will become both are dominant. Okay, capital R, capital Y, capital Y. So it will become round yellow. Again here, round. In between these two, don't mean confusion. Capital Y, small y means small y is the dominant. No need to write the green because the capital Y is the dominant one. You have to mention which is the dominant. That is the yellow. Then again, same here, round yellow. Then again, in between these two, capital R is the dominant. Okay, so it will become round yellow. The next, they both are dominant, so it will be round. In between these two, small y is there, it is recessive, so it will become yellow. Round here, both are recessive. Small letter, written in small letter. So they both are recessive means then we have to mention. Okay, so it is round green. Okay, the next round yellow. Then round green because small y both are recessive. Round green. Then again come to here, round yellow. Round yellow. Then smaller. What it will become? Wrinkled. Okay, it is wrinkled yellow. Wrinkled yellow. Okay, the next is round yellow. Here it is round green. Here it is wrinkled yellow. And lastly it is wrinkled green. Now we have to find out the ratio. So let's see about the ratio. Here I will show you. See, phenotype ratio. Next is genotype ratio. What is phenotype ratio? Means we have to see the external characters. That means we have to see only the color and the shape of this. Okay. If we see like that means round yellow 1. Okay. How many round yellow are there? 1. 1. Round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, round yellow 6, round yellow 7, 8, then round yellow 9. How many round yellow are there? 9. Here I will write round yellow, that is 9. Okay, then next round green. Round green 1, round green 2, then round green 3. How many round green are there? Round green, that is 3. Okay, here how we have to write means by applying a mathematical expression, that is ratio. Okay, 9 is to 3 is 2. Then come to here. Wrinkled yellow. This is the third character. Wrinkled green 1. Sorry, wrinkled yellow 1. Wrinkled yellow 2. Wrinkled yellow 3. Okay. Here 9 is to 3 is to. That is wrinkled yellow. Wrinkled yellow 3. The last tree, only one is there, that is wrinkled green. So here we can write wrinkled green 1. So what will be the phenotype ratio? 9 is to 3 is to 3 is to 1. Okay. The next, come to the next one, that is genotype ratio. Genotype ratio means we have like that. This is 1. Then the genes should be same. Capital R, capital Y, small y. 1 is there. 1. This is the 2. Then 1, 2. Only 2 are there. So we can mention here 1 is to 2. Okay. Then third one. Capital R, small r, capital Y. Like this, how many are there? This is 1. Then this is 2. 2. Okay, 2 are there. Okay. 1 is to 2 is to 2. The next one. Capital R, small r, capital Y, small y. 1, 2, then 3. Okay, 1, 2, 3. Then here 4. 1, 2, 3, 4. Okay. 1 is to 2 is to 2 is to 4. The next, come here. Capital R, small y. 1. Then next comes only 1. Like this, only 1 is there. 4 is to 1. The next, capital R, small y. 1. Then here it's a 2. Again 2. 2 is 2. Then small r, capital Y. 1. Only 1 is there. 1. Next, small r, capital Y, small y. 1. Then 2. 2. Lastly, this is the 1. Okay. So this is about the genotype and phenotype ratio of what? Dihybrid cross. The next, we will go for the next topic. So this is very important. That's why I had repeated once again. The next, we will go for the law of independent assortment. Next, law of independent assortment. So let's see here. After completion of dihybrid cross, Mendel observed some of the law, or they had generalized one law, and he named that law as the law of independent assortment. What does it state? Let's see that one. Law of independent assortment. Based on the Mendel's completion on the dihybrid cross or work on the dihybrid cross, he had invented a law, that is law of independent assortment. Okay. So here, what does it state? Means this states that factors for two or more pairs. This law states that factors for two or more pairs of two or more pairs of contrasting characters two or more pairs of contrasting characters are distributed independently of one another. Are distributed independently of one another at the time of gamete formation. At the time of gamete formation. Okay. Now listen here. What this law states means, this law states that factors, that means the genes which is for two or in the dihybrid first we had talked about the two characters. What are the two characters? That is for two characters or more than that also we can see. What are the two characters here in the dihybrid? What we had studied means, the two characters are shape of the seed, and the second is color of the seed. Color of the seed. These are the two characters of what? Of that particular genes. Here, what are the genes? Capital R, capital Y, small r, small r. Okay, these are the two contrasting characters. Two or more than that also. Okay, what I am explaining here is, here factors for two or more pairs of contrasting characters. What do you mean by factors? In the Mendel's language, we can call these factors as the genes. Sorry, genes as the factors. Okay, so here the factors for two or more than that contrasting characters are distributed independently. What it is? It is distributed independently of one another at the time of gamete formation. See, if we, these are the two characters. What are that? Shape of the seed, what we have taken? That is round and wrinkle. These are the two contrasting characters. And color of the seed means what are the two colors we had taken here? That it is yellow and green. That are the two characters what we have taken in the dihybrid cross. Okay. In that, what was the parents? Here you see the parents was round yellow. This is one parent, and one more parent, that it is wrinkled green crossed with wrinkled green. So these are the two characters. Round is the shape of the seed. Round and wrinkled. These are the two opposing characters. Okay. Yellow and green. These are the two opposing characters or two contrasting characters. What they are saying in the law of independent assortment means two or more pairs of characters that is going to distributed independently during what? During the gamete formation. Here you see, these are the factors. What are that? Capital R, capital Y, small y. These are the factors or genes crossed with small r, small y, small y. See, for round, this is the factor. For yellow, this is the factor. For wrinkled, this one, and for green, this is. So, these factors which are present in pairs, that is going to independently separated when the gamete formation is going to take place. Here, this is the gamete formation. Okay, during gamete formation, what happens? See, gamete form. What are the gametes here? In the male flower, it will be the pollen grain, and in the female flower, it will be the ovule. During that formation, the parents are going to transmit their genes or factors for a particular trait or character. Okay, so here this is going to separate. Okay, they are not going to transmit or inherit in pairs. That is going to separate capital R and capital Y. Like this. They are going to separate. Why? Because there is a child which is going to take place. That I will further explain. Okay. Then here also, from these two, one pair, one is going to separate it and another is going to separate it. This only explaining in what means, that is in the law of independent assortment. Okay. So this is about what? Law of independent assortment. So here in F1 generation, what we had got? We had got, if you cross like this, we had got capital R, capital Y, small r. After this, in F2 generation, for that the parents, what are the genes for the parents? That it is capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the gametes for what? For F2 generation. Okay, so these are also separated during the gamete formation. Okay, so this is about what? About the law of independent assortment. The next we can go for the next topic, that it is the chromosomal theory of inheritance. Next it is chromosomal theory of inheritance. This is the next topic, that is the chromosomal theory of inheritance. See, first thing, Mendel's knowledge about genetics or the genes which is going to pass from one generation to another generation was indispensable, means was not accepted more. Okay, here, so Mendel first, when he published his work on inheritance, that is in the year 1865. Mendel published his work on inheritance of character. On his work on inheritance of character. Inheritance of character, that is in the year 1865. In the year 1865. But what happens? First he published his work on inheritance of character in the year 1865. So, but for several reasons, it was remain unrecognized. Okay, he has not got so much of success on what? On this genetics or on this inheritance of characters. Okay, so he has it, but for several reasons. His work was remained unrecognized. His work was remained unrecognized. Till which year? Till 1900. He published his work, but it was not got so much of success. Okay, because most of the person don't know what is genetics, what is going to inherit. They had disproved it. Okay, so till 1900 it was unrecognized. But firstly, what happens? Communication was not easy at that time in 1900s, and his work could not be widely published in a narrow range. It is not widely published. Secondly, his concept of genes. For his concept of genes are the factors. His concept, the first concept, what it was means. Here you see the first concept on genes. Genes, which is also is going to call it as the factor. This concept on genes or factors in Mendel as stable and discrete units. What he told, these genes or factors are discrete units that control the expression of traits. What he told, these genes or units, these are the discrete units which controls the expression of traits. Which controls the expression of trait. Trait means what? That is the carry. Okay, this is the first concept. Then second concept, what it is means, in that trait he had mentioned as one of the things that it is the alleles. Okay, so the pair of alleles which do not blend with each other was not accepted by the contemporaries as an explanation of inheritance. Okay, see here, alleles which do not blend. Alleles means, as I explained in the last class also, what do you mean by alleles? If same chromosomes are there and there is a presence of a pair of genes, okay, that we can call it as the alleles. They are not going to mix up. Okay, that alleles are not going to mix up. So alleles does not blend. Okay, this is the second concept. Do not blend with each other. Means they are not going to mix up with one another. Okay, see, if this is one gene on one chromosome, and these chromosomes are the homologous chromosomes. Homologous means which are present at the same location. So that we can call it as the homologous chromosome. So these genes are not going to mix up. Okay, so it was not accepted. This thing is not accepted. Not accepted. This explanation was not accepted, and so it is not the what they told. They had been disproved this explanation on what means allele does not get blend expression. And what is that mathematical expression? That while after that, whatever the phenotype ratio, genotype ratio you have to find out, in that we use the ratio. So this is also somewhat unsatisfiable. Okay, so mathematics to explain biological phenomena was totally new and unacceptable for many biologists, because in biological they are going to apply, Mendel has been applied in this biology mathematics. So many of the biologists has been unaccepted or disproved this also. Okay, this mathematical to explain the biological phenomena, it was totally wrong, and it is a new concept to many of the biologists in that time. Finally, the Mendel was suggested that factors was discrete units which could not provide any physical proof for this existence of what? Inheritance of characters. So for this reason, due to these concepts, three concepts, one is gene, second is allele, and third is applying a mathematics to a biology. Due to these three concepts, his work remained unrecognized. Then some more scientists worked on this inheritance of chromosome or inheritance. So they had proved that one. They have proved, and they got the success on Mendel's work. So next we will continue in the next class.